Now, today we are going to separate a mixture of two dyes, the rosaniline hydrochloride, which is a red dye, and bromophenol blue, which is a blue dye. So we'll be giving you a one-to-one mixture of two dyes. We'll be mixing them together, and we'll be asking you to separate those two compounds, two dyes. Now, any technique that you use in organic chemistry to separate the components of a mixture, it's called chromatography. We are doing chromatography, means you are separating the components of the mixture. Oftentimes when you do a reaction, you may not get pure product, so there will be a main product and also there will be something else as well. You need to separate them, and many times in organic chemistry labs you have to separate them. So you use two techniques, very frequently used techniques, thin layer chromatography and column chromatography. Thin layer chromatography is known as TLC. Thin layer chromatography is a very common technique used in organic chemistry labs. So what happens is, in TLC, you take a strip, a glass back strip or plastic strip, which is a coating of some solid phase or stationary phase. Stationary phase usually is silica gel. It's a silica gel, or you could use alumina as well. Alumina is used less frequently. Silica gel is used more frequently. But today, in this lab, you'll be using both of them. You will be using alumina as stationary phase in column chromatography, but silica gel in the thin layer chromatography. So thin layer, we call it thin layer because the thin layer of the stationary phase silica. So it is kind of a smear on the surface of the glass plate. You call it TLC plate, and it's commercially available. Basically, you know how to prepare them. So what you do is basically you take a TLC plate and you take a number 2 pencil and draw a very lightly line, horizontal line parallel to the lower side of the TLC, about 1.5 centimeter from the bottom to here. And you also draw a line using a ruler and number 2 pencil, about 1.5 centimeter from the top face as well. So now basically you are ready to spot. Well, spotting means that you take a mixture and you dissolve it. In fact, you'll be given mixture in already a solvent. So you just basically use a capillary. It's a small capillary tube, and you dip your capillary tube in the mixture. The mixture rises up in the capillary, and then you bring it over here and very lightly spot right middle, somewhere in the middle here. That's the mixture over here, right? So that's the spotting the mixture. Now, what about these two, red and blue fractions? Since you do not have red and blue fractions right now, so you basically have only one spot here, which is the mixture. Now, these two you will be getting after separation. So you will be separating the red, rosaniline hydrochloride and bromophenol blue, using this column chromatography technique. In column chromatography, it's usually a tube, and you will be using actually a posture pipette as a tube, as a column, right? A column, So, columns that will be as big as this one. We have a very small column, like a pipette that can also be a column. Today you'll be using a pipette as a column. So an empty pipette, and add a little bit of cotton at the bottom, and then you basically fill with the alumina powder. Alumina powder is very free flowing, so you can actually easily add it even into the pipette using a weigh boat. And then at the here, on top of that, you add a little bit of sand. Okay, so sand actually keeps this alumina powder between the two limits, between the cotton plug and the sand. It's a compact column. All right, so now you are ready to saturate this column with the mobile phase. Mobile phase is also called eluent. Okay, eluent or mobile phase. It's a 95% ethanol. So what you'll be using is basically adding 95% ethanol with a pipette, and you will wait till the solvent moves, eluent moves through the column and collects in a tube. We call it just a P. P means pass through, just an empty tube. So right now there's no mixture, right? There's basically no mixture at all. It's just basically just alumina and sand and a cotton plug. Now you are saturating the column with the solvent, the eluent. Okay, so you basically collect about, I would say, 5 or 6 mil of eluent. Then you kind of assume that this column is saturated with 95% ethanol. Okay, now you are ready to do the separation. Now you have, for separation, you have to load, we call it loading, loading a few drops of aniline hydrochloric chloride and bromophenol group mixture. But before you do that, you're already actually putting in 95% ethanol, and it's coming down. When the 95% ethanol hits the sand, you immediately add 2 or 3 drops maybe, add on top of that, and then wait till it sinks into the sand. Okay, as soon as it sinks into the sand and into the column, then you immediately load it with more 95% ethanol in this blank area. Okay, so make sure at no time this column should run dry. As soon as the level of the solvent or eluent, the mobile phase, as soon as it gets close to this sand, add more, right? So you keep adding more and more. So you have to be very vigilant about this. Keep adding more, and what will happen is here you have the dye mixture as well. So dye mixture now will start separating. You will see that the red is moving faster than the blue. In fact, the blue probably doesn't move or moves very little. Okay, but red rosanilin hydrochloride, which is red color, it comes running down basically very quickly. So when right now it's a pass through means a blank tube basically, right? When the red color here is coming down, right? When it reaches this point basically, then you change the tube with R. R means red. So you have another tube which is labeled R. You insert it over there. Now you collect all the red color. Okay. Now red color basically, when you can tell red color is all out. When it's all out, then you change the solvent. Instead of 95% ethanol, then you add water. Just water. So water is the mobile phase now. So there are 2 mobile phases in column chromatography. One is 95% ethanol and the other is water. Okay. Now you have water. Water is more polar, and also the blue dye is also more polar. And so now the water will actually bring it down. So you see the blue color moving down right here. And when it reaches here, then you basically change the. By the way, when you have or red color is all out, you remove the tube and put the pass-through tube back again. Okay, because there's no dye coming now. Okay. Now when the blue dye comes in, when it gets close to the cotton plug, then you change this tube with the blue tube. Okay, with the blue tube. Now you collect that fraction as well. So now you have 2 fractions, one red fraction which is in the R tube and one blue fraction in the B tube, and so basically you want to concentrate them, because if you don't concentrate them you will not see any spots. So you need to concentrate them using compressed air, and the instructor will show you how to do that. Okay, so when you have concentrated a little bit bigger volume but a very small volume, then you can take a capillary and dip into the red or blue tube, not tube, concentrated solution, right? And spotted here, red here, and blue there. Now you have three spots. Red spot, which is the red fraction that you separated from the column chromatography, and in the middle, the mixture, which was not separated, unseparated mixture. That's also a spot over here. The third spot belongs to the separated blue. Okay. All right. So you have three of them. Now you put it in a TLC chamber. Okay, and you cover the chamber. Make sure you put it in like a slanting position. Now the solvent will rise from the bottom and it will go up. See, the mobile phase is moving up in TLC and the mobile phase is moving down in column chromatography. But the principle is the same. The principle is that the dyes are different, they have different structures, they have different mobility in the solid phase. Here the solid phase is a layer of silica gel. Here you have alumina powder, a column, that's a solid phase. Okay. So what will happen is that as dyes have different structures, they have different mobility, they have different polarity. Okay. So depending upon the difference of polarity, you can actually separate them. If the polarities are very similar, it will be very difficult to separate. Okay. So only those compounds that are they show some polarity differential between them, those are easy. The bigger the difference in polarity, the easier it is to actually separate the compounds. Here you can visually see red and blue, but many times, in fact most of the time, the compounds that you separate, they are colorless compounds, so you have to actually visualize them using ultraviolet light or using some kind of stain. Okay. Alright, so that's those are the two techniques that you'll be using. First you'll be using column chromatography, and then you will be analyzing the fractions, that the red and blue fractions, and the mixture on the TLC plate. Now, you might see the difference between the results. Here, the first color that comes out is the red color, and the blue stays behind. Here, the blue comes first, and the red stays behind. And you have to watch my PowerPoint presentation, where I have tried to explain to you why there is a difference between two stationary phases. Okay, so let's get to the code and do the experiment. Okay, we're back in the lab, and this is the posture pipette that I'm holding in my hand. And let me introduce to you the things that we need for this experiment. This is a TLC chamber where we put our thin layer chromatography plate, TLC plate. And this is the mixture that we have to analyze and we have to separate. This is rosanilene hydrochloride and the bromophenol. Bromophenol blue, rosanilene red is red. Now we want to use the TLC plate. We want to cut the TLC plate. TLC plate comes in 20 by 20 centimeter sheets. They are commercially available, and they are on one side they are coated with either silica gel or with alumina. In this case, they are coated with silica gel. And we want to make sure when you want to make TLC plates, cut TLC plates, we want to mark them with a pencil, number 2 pencil. Hold the tool pencil very lightly and cut the 20 by 20 centimeter plate into 4 parts, and then you can actually cut them with a pair of scissors. So make sure you very lightly, about 1.5 centimeter from the bottom, make a line with a pencil, very lightly. Don't put too much pressure on the pencil. Also from the other side, 1.5 centimeter on both sides. Okay, don't use too much pressure on the pencil when you are. So I have done this, and I have now marked 3 points on this lower line. The lower line is called baseline, and the first this is the
alumina absorbent or solid phase. That's what we're going to be using. Okay, so this is the same alumina. We are going to use alumina for our column chromatography. So we have to prepare a column. We take a pasture pipette, 9-inch pasture pipette, and we want to put a small plug of cotton so that our alumina cotton can be pulled up from a cotton swab. Use a cotton swab stick and remove a little bit of cotton from there. You can pull it out with the fingers, and you want to put it inside the pasture pipette. You can use the stick to push it down all the way to where it stops. Now it's very important that you do not put too much pressure. Just very lightly, all you want to do is just block that passage. Okay, very lightly. Don't put too much pressure. Otherwise, what will happen is the cotton will be too tight and it will block the flow of your solvent later on. Now you can actually pinch your whey boat that has alumina and start pouring it very carefully into your pipette, and then you add on top of that a little bit of sand, only a little bit. So most of it is alumina, but only a little bit would be a layer of a sand. You can layer some sand on top of it. So this sand is beach sand, and so I have prepared this column. As you can see, this is a pasture pipette, uh, and I have poured the aluminum a little bit below the groove, and on top of that you can see very easily a layer of sand. Now we want to saturate this column. This pasture pipette is I call it column now. It's a column chromatography. Okay, even though people use bigger columns, but for this lab we use only a very small column. Pasture pipette is the column. So you now load it with the solvent. Solvent is 60% ethanol, 40% water. That solvent is provided to you, and you add solvent all the way to the top. Make sure that the reservoir is almost full. Reservoir means the space above the sand. Okay, so as you can see, it's going down now slowly. Okay, so we know that the solvent has been going down. Now it's very close to the sand. So now we can add or load the column with the dye mixture, the rosaniline hydrochloride and bromophenol blue, and then let the dye sink into the sand first, and then into the alumina column, and then load a little bit rinse of ethanol. Don't use 60-40% anymore. Just use ethanol now, and with the ethanol, wash it down all the way to the a little bit few drops all the way to the sand, and then onto the column. Okay, and then add a little bit more. A couple of rinse should be used with small amounts of ethanol. And when we are sure that the all the dye mixture has sunk in, then we can actually add more solvent all the way to the top. Okay, so we have filled the reservoir. Remember, reservoir is the space above the sand. Okay, so we are back in the lab. As you can see now, I have been loading the solvent into the reservoir on a regular basis, and so you can see the dyes are separating very nicely. The red color, which is rosaniline hydrochloride, you can call it red or magenta color, it is coming down quicker. And notice that I have, this is the R means red. Previously it was P. P means pass through. The pass through was clear solution, no dye in there. Now I am actually collecting the red dye, which is slowly moving down. So the mobility of red color is faster than the blue color. So I'm back in the lab, and I have already collected the red color xanilin hydrochloride with the 95% ethanol. So 95% ethanol has done its job. It has separated the red color in the vial R, and I substituted the vial with pass through again, and now I'm going to use the other eluent, water. Water will elute the blue color, and blue color will slowly be eluted just like the red color. At that point, when it starts coming, then I will switch it with the blue vial. Until then, so as you can see, the blue color is coming down now. It's being eluted with water, the second eluent. And I have used the pass through because the blue color is not coming out yet. So when it starts coming down, I will be switching it with B, bromophenol blue. It's almost there now, so I'm going to change pass through vial with the B vial, bromophenol blue. Now I'll be collecting the blue color. We are now back in the lab. Now we can see that the blue dye is also coming down. It's also being eluted and into the vial B. Now we should actually concentrate the red eluate by pressurized air. Make sure that when you do that, you adjust the airflow. At the same time, you should keep a watch on the eluent as well. Make sure that the eluent should remain uh, in the column all the time. It should never be allowed to run dry. So you should actually concentrate the red dye by using a little bit of heat and compressed air. Now let's talk about the TLC that we prepared last time. We drew two lines, one from one in the one in the baseline and the other from the top. Now you take a capillary. Capillary is open on both ends, and this is the one that actually picks up the sample through capillary action, and we can spot it on TLC. So make sure you should add more solvent onto the column. The column should never be allowed to run dry. And this time I can tell you that we are getting almost to the to complete the separation of the blue dye. We had already separated the red dye. Now we are doing we are separating the blue dye. Now it's difficult to get the sample, not difficult, but a little bit clumsy to get the sample from there. So you make sure you dip the capillary in there, and you should be actually using a 3-well plate to concentrate the sample. Also you should transfer the blue dye over there. Okay, now you should with the help of capillary, you should spot on the left side. There are 3 locations. On the left, I marked them: left, right, middle, and right. On the left side, you should basically very lightly touch on the coated surface here. Okay, you can see the red dye over there. You can after about 30 seconds, spot it again exactly at the same time. Exactly at the same time. Make sure that you are very careful about it. You should spot it very carefully immediately. Do not keep the capillary on the layer. You should just touch it and remove it. The size of the spot should be as small as possible. Okay, we can do the same thing. Now our elution of the blue dye, bromophenol blue, is also complete. Now we should concentrate it with heat and compressed air exactly the same way, uh, the way we did the red dye. Now basically our experiment is complete. Uh, the column chromatography is complete. Now we are concentrating the dye using heat and compressed air. Again, we can uh, we should transfer the blue dye on the 3-well plate. You can see that on the right side, and from there you can concentrate more. I think this blue dye must be concentrated quite a bit. You should reduce the volume quite low. Now capillary can be, you see there's a hole in there. You can actually, through that hole, you can pour pour capillaries. All right, so this is the reaction. This is the mixture that we were originally given to separate. So this is a sample. It's a 1-to-1 dye. You should also spot exactly the same way. Since this dye is already concentrated, you need to spot it only once. The others you need to multiply spot it like 2 or 3 times. Now this is the filter paper that needs to saturate the solvent, the vapor, the purpose of this filter paper is to keep the environment inside the TLC chamber saturated. So that helps to saturate the environment very quickly. All right, so this is our TLC chamber. We need to put the solvent in there, and make sure when you put the solvent should be 60% ethanol, 40% water, and you should add solvent to the chamber, wetting the filter paper first, wetting and adding about 1 centimeter from the 1 centimeter from the bottom. Okay, do not add too much. If you add too much, then your spots will be dipping into the solvent, and that will ruin your experiment. So it should be about 1 centimeter. Okay, so you can actually measure it. It's a visual inspection. Your spots should be higher than the liquid layer. It's very important. So we are going to place the TLC plate into the TLC jar with the spots pointing down, and we are ready to put it now. By the way, the jar has been there for about 10 minutes, so the filter paper is already wet. So you place the TLC plate inside the jar on the opposite side of the filter paper. Again, you want to make sure that the spots do not dip into the spots should not be dipping into the solvent. Okay, you basically put the top onto the jar. You close the jar, and now you wait till the solvent will slowly rise up. As it rises up, it will take the spots with it, depending upon the properties of the dyes. Some dyes are more polar than the other. The less polar will go up, and the more polar will be lagging behind. So we are in the lab, and I have been actually waiting for the last about half an hour, and still the solvent has not risen all the way to the top line. So I can see the separation of the spots, so I removed the plate, TLC plate, from the jar. I can see that the blue dye and the red dye have been separated. However, the solvent front has not moved all the way to the top. It has moved only where I'm drawing the line. Okay, you can draw the line with the help of a ruler or just by hand. And so note down the middle of the spots, the thickest part of the red spot and thickest part of the blue spot, and make sure you mark them with a pencil. The thickest parts of the spots should be marked with the pencil. And this the middle spot was a pure blue spot which was isolated. It's very light. That means that the concentration of the blue dye was not very good. So I suggest that the original solution should be prepared in such a way there should be higher concentration of blue compared to the red. Red is not a problem, blue is the problem. So use about 3 parts of blue and 1 part of red in ethanol to make a solution, and then the TLC should give you very clear spots here, right close to the top line. And so this, you can see that the solution is very dilute, and it didn't show up very nicely on the TLC plate. But you can see it. You can find out the RF value. First of RF value, you need to mark the middle point, the thickest part, with a pencil because you need it for RF value. Now measure the distance between the top line, between the baseline and the middle of the spot. That's distance A, and then here all the way to this front line is distance B. A divided by B will give you the RF value for the red spot. Same thing for the blue spot. The distance between the baseline to the middle of the blue spot is A divided by the distance between the baseline to the solvent front is B. In this case, the RF value is almost 1. You remember that last time the blue spot went all the way to the front line, and that was bad because RF value was almost 1. RF value should be always less than 1. So I've been trying to actually find out a proper solvent system where I can run my TLCs. So I have done 3 experiments. One experiment was done in pure ethanol as eluent, not 60%, 40%, but pure ethanol. The second one was done in 95% ethanol. Third one was 80% of ethanol and 20% acetone. And it turns out that all the 3 solvents that I used, they gave me very similar results. So ethanol is very expensive. 95% ethanol is cheap, and therefore the middle one, 95% ethanol, is what I recommend basically for running the TLC instead of 60 to 40% as in the manual. So now why this spot is not green? Why it is yellow? Well, do you remember that this is a pH, this is a bromophenol blue, which is pH sensitive. At pH 3 it's yellow, and at pH beyond 3, between 3 to 4, it's blue. So silica gel is acidic, and that's why it reduces the pH from 4 to 3. That's why it's looking yellow. So why these spots are oblong? Why they're dragged? Because these are acids, and acids are very highly polar, and they drag on always on TLCs. They give you oblong, not really round spots, but oblong spots. But you get to measure the RF value because you just measure the distance from the baseline to the middle of the oblong. Okay, now the RF value for the green spot, or we have started from RF value is distance A divided by distance B, and same thing with you take the middle point of the red point, right? Distance A would be from the baseline to the thick part of the spot, and divided by the distance between the baseline to the top solvent front. Now these green have the same RF value, and these two red have the same RF value.